Omics is a revolutionary approach that allows us to study biological molecules at an unprecedented scale. It provides a comprehensive, holistic view of biological systems, transforming our understanding of genetics, diseases, and ecosystems. Omics forms the foundation of precision medicine and modern biotechnology, enabling us to make groundbreaking advancements in these fields. The image on the slide depicts a state-of-the-art scientific laboratory, where researchers are analyzing genomic data and visualizing complex DNA structures on large screens. This cutting-edge facility represents the kind of high-tech environment where omics research is being conducted. Omics is the collective characterization and quantification of pools of biological molecules that translate into the structure, function, and dynamics of an organism or system. Omics studies aim to reveal the complete sets of molecules, such as genes, proteins, and metabolites, and how they interact to make up the fundamental processes of life. The omics terminology denotes fields that study complete sets of molecules, like genomics, proteomics, and metabolomics. The OME refers to the objects of study, like the genome, proteome, and metabolome. Omics approaches provide a more holistic understanding of biological systems compared to studying individual molecules or pathways in isolation. The slide provides an overview of the evolution of biological research, highlighting 3 key stages. The first stage is the traditional approach, which focused on studying individual components in isolation. The second stage is the technological revolution, which enabled the development of high-throughput methods for system-wide analysis. The final stage is the omics approach, which allows for comprehensive analysis of entire biological systems at once. The timeline visually depicts this progression, with each stage represented by a numbered, 2-sided timeline element. The slide is accompanied by an image that visually represents the evolution of scientific research equipment, from a basic microscope to an advanced DNA sequencer. Omics is a powerful approach that allows us to study entire biological systems in a comprehensive way, rather than just looking at isolated components. By examining the interactions between all the different molecules and components, we can gain a much deeper, systems-level understanding of how these complex systems function. Omics integrates data from multiple molecular levels, genomics, proteomics, metabolomics, and more, to build cohesive, multi-layered models of biological processes. The massive datasets generated through omics technologies enable us to identify important patterns and make new discoveries that would be impossible through traditional, hypothesis-driven research alone. Omics technologies have opened up exciting new frontiers in scientific discovery. They allow us to reveal previously unknown relationships between different molecules in the body. Omics provides important context for understanding how individual molecules function within the larger biological system. This contextual understanding enables us to make more accurate predictions about complex biological behaviors. Omics data is also proving invaluable for identifying promising new biomarkers and therapeutic targets. Next-generation sequencing allows us to rapidly and cost-effectively sequence DNA and RNA, providing unprecedented insights into biological systems. Mass spectrometry is a powerful technique that can identify and quantify proteins, metabolites, and other biomolecules, giving us a detailed view of the molecular composition of cells and tissues. Nuclear magnetic resonance spectroscopy provides information about the structure and dynamics of molecules, complementing the data we get from sequencing and mass spec. Bioinformatics tools are essential for processing and interpreting the massive datasets generated by these advanced technologies, turning raw data into meaningful biological insights. Together, these cutting-edge technologies are enabling a revolution in our understanding of living systems at the molecular level, the field of omics research. Genomics is the foundation for understanding the human genome and how it functions. DNA sequencing allows us to read the complete genetic code, revealing the building blocks of life. Analyzing genetic variation helps identify differences between individuals and how those differences may impact health. Functional studies determine the specific roles and functions of genes, shedding light on biological processes. Applying genomic insights in clinical settings enables more personalized and targeted approaches to medicine and healthcare. Genomics is the study of the complete set of genetic information in an organism. There are several key types of genomics research, each with a different focus and approach. Functional genomics looks at gene functions and how they interact in regulatory networks. Comparative genomics compares genomes across different species to find similarities and differences. Cognitive genomics studies the genetic factors that impact brain function and cognition. Structural genomics analyzes the three-dimensional structures of genomic elements like genes and proteins. Each of these areas provides valuable insights into the complex world of genetics and biology. Transcriptomics is the study of the complete set of RNA transcripts produced by the genome under specific circumstances. It reveals which genes are actively expressed in different cells and conditions. Key techniques in transcriptomics include RNA sequencing, microarrays, quantitative PCR, and single-cell transcriptomics. The image on the right shows a visualization of RNA sequencing data, with a colorful heat map display representing gene expression levels in a molecular biology laboratory setting. Proteomics is the large-scale study of proteins, their structures, functions, and interactions within cells and organisms. The proteomics workflow involves three key steps: sample preparation, separation and analysis, and data interpretation. In sample preparation, proteins are extracted from cells or tissues and undergo purification and processing to prepare them for analysis. Mass spectrometry is a core technology used in proteomics to identify proteins by their mass and charge. Additional techniques can reveal detailed structural information and post-translational modifications. The complex datasets generated through proteomics analysis require sophisticated computational tools and bioinformatics approaches to map the proteome and uncover patterns, networks, and biological insights. Metabolomics is the study of metabolites, which are the small molecules involved in cellular processes. It reveals unique patterns of metabolites that can serve as chemical fingerprints for different conditions. Metabolomics provides an immediate snapshot of cellular activity, giving a real-time status of what's happening. This can be used to identify biomarkers, indicators of disease or treatment response. Understanding these metabolic patterns can lead to important discoveries about health and disease. Metagenomics is the study of genetic material collected directly from natural environments, rather than from individual organisms. The key steps in a metagenomic study are environmental sampling to collect genetic material from a natural setting, DNA extraction to isolate the genetic material from all organisms present, sequencing to read the combined genetic information, bioinformatic analysis to identify the species and functions present in the sample. Metagenomics allows us to study the diversity and functions of entire microbial communities, rather than just individual microbes. This provides valuable insights into the ecology and roles of microorganisms in natural environments. Microbiomics is the study of the collective genomes of microorganisms in specific environments. It reveals how microbial communities impact areas like health, agriculture, and ecosystems. The slide shows visualizations of different microbiomes: the gut, skin, soil, and ocean. These diverse microbial communities play critical roles in their respective environments. Understanding microbiomes is an important area of scientific research with many real-world applications. Epigenomics is the study of chemical modifications to DNA and chromatin that can turn genes on or off without changing the underlying DNA sequence. DNA methylation is a key epigenetic process where methyl groups attach to DNA, which can silence gene expression. Histone modifications involve chemical changes to the histone proteins that DNA is wrapped around. This affects how tightly the DNA is packaged, impacting gene accessibility. The overall chromatin structure, how DNA is organized and packaged in the cell nucleus, also plays a key role in regulating gene expression. Understanding these epigenetic mechanisms is crucial for studying gene regulation development, and disease. Phenomics is the comprehensive study of phenotypes, which are the observable characteristics of an organism. It involves measuring and analyzing phenotypes at different levels, from the molecular to the whole organism. The key measurement techniques include metabolite profiling, proteomics, flow cytometry, medical imaging, and physical measurements. These techniques have diverse applications, such as biomarker discovery, drug screening, disease diagnosis, and crop improvement. Phenomics provides a holistic understanding of biological systems and enables the development of new therapies, diagnostics, and agricultural technologies. The human lipidome is estimated to contain over 10,000 distinct lipid species. Lipids make up about 30% of the total weight of a typical cell membrane. There are 5 major categories of lipids: fatty acids, glycerolipids, glycerophospholipids, sphingolipids, and sterols. Lipids play a crucial role in the structure and function of cell membranes, serving as structural components and signaling molecules. Understanding the diversity and distribution of lipids in the human body is the focus of the field of lipidomics. Interactomics is the study of all the interactions that occur between different molecules within a cell or organism. The pie chart shows the breakdown of different types of interactions that have been identified through interactomics research. Protein-protein interactions make up the largest portion, at 45%, reflecting the central role of proteins in cellular processes. Protein DNA and protein RNA interactions are also well represented at 25% and 15% respectively, highlighting the importance of gene regulation and expression. Metabolite protein interactions account for 10%, demonstrating the integration of small molecule metabolism with protein function. The other interactions category, at 5%, likely includes more specialized or less common types of molecular interactions. Understanding this interactome landscape is crucial for unraveling the complex networks that govern biological systems. Multi-omics integration provides a comprehensive understanding of biological systems by combining multiple omics datasets like genomics, transcriptomics, proteomics, and metabolomics. Integrating these diverse data sources allows us to gain deeper biological insights that would not be possible from analyzing any single data type alone. Advanced computational algorithms and methods are required to effectively analyze and interpret this complex, cross-platform data. The goal is to uncover meaningful connections and patterns that reveal the underlying mechanisms driving biological processes and disease. Bioinformatics is the backbone that enables the powerful omics fields like genomics, proteomics, and metabolomics. It involves critical data processing steps like quality control, normalization, storage, and efficient retrieval. Advanced analysis methods are used, including statistical tests, pattern recognition, pathway analysis, and network modeling. These techniques unlock insights from the vast amounts of biological data being generated. Cutting-edge applications leverage machine learning, AI, predictive modeling, and systems biology approaches. Bioinformatics is essential for translating omics data into meaningful biological discoveries and innovations. Omics research, like genomics, proteomics, and metabolomics, is generating massive amounts of complex biological data that is challenging to manage and analyze. The sheer volume of data, often reaching petabytes, requires specialized infrastructure and computational power to store, process, and extract insights. Living systems are incredibly complex, with countless interacting components and nonlinear relationships that make it difficult to fully understand biological processes. Diverse experimental protocols and data analysis platforms create compatibility issues, making it hard to integrate and compare results across studies. There are important ethical concerns around personal biological data, including privacy, consent, and ownership that researchers must carefully navigate. Precision medicine uses omics data to tailor treatments to a patient's unique genetic profile, improving efficacy and reducing adverse effects. Multi-omics research reveals complex disease pathways, allowing researchers to identify molecular drivers of disease progression and develop novel therapeutic targets. The image shows a doctor reviewing a patient's personalized genomic profile, highlighting how this technology is being applied in a modern medical setting. Agriculture is a key application area for omics technologies. Omics can accelerate crop breeding programs by identifying beneficial genetic traits. Genomic insights help develop crops that are more resilient to diseases and pests. Multi-omics approaches enable the creation of crop varieties that can thrive in challenging environmental conditions, like drought. These advancements in agriculture are critical to meeting the growing global demand for food in a sustainable way. Environmental science has many important applications that help us understand and protect our natural world. One key application is ecosystem monitoring, where we track biodiversity and the overall health of ecosystems over time. Another is pollution assessment, which involves detecting contaminants in the environment and studying their impacts on living organisms. Climate change studies help us understand how species and ecosystems are responding to changing environmental conditions. And conservation strategies rely on scientific data to inform efforts to protect endangered plants and animals. These diverse applications of environmental science are critical for making informed decisions to safeguard our planet. The emerging omics fields we'll be discussing today offer powerful new ways to analyze biological systems at unprecedented resolution. Single-cell omics allows us to peer into the molecular profiles of individual cells, revealing the hidden heterogeneity within tissues. Spatial omics takes this a step further, mapping molecular information to specific physical locations, providing a detailed spatial map of biological processes. Exposomics examines the lifetime environmental exposures that shape our biology, opening new windows into the drivers of health and disease. And foodomics explores the molecular-level impacts of food components, helping us understand the intricate connections between diet and well-being. These cutting-edge fields are transforming our understanding of biology and ushering in a new era of precision medicine and nutrition. The future of omics technologies is exciting, with several key directions that will shape the field. Deep integration will allow us to seamlessly combine different omics layers, genomics, proteomics, metabolomics, and more, for a more holistic understanding. AI-powered analysis will be crucial to extract meaningful insights from the increasingly complex omics datasets we're generating. Miniaturized, point-of-care omics devices will bring these powerful technologies out of the lab and to the bedside or even remote field settings. Ultimately, the goal is to democratize access to omics technologies worldwide, making them available to everyone who can benefit. Personal omics data contains sensitive information about a person's health, genetics, and other private details that could be misused if not properly protected. There are questions around who owns this biological data, the individual, the company that collected it, or others. Clear policies are needed on how this data can be accessed and used. Explaining the complex science and potential implications of personal omics technologies to research participants is challenging. Ensuring truly informed consent is critical. We must work to make these powerful technologies accessible to all populations, not just the wealthy, to avoid exacerbating health disparities. Omics is an interdisciplinary field that combines biology, computer science, statistics, and data visualization. Students need a strong foundation in biology fundamentals, as well as skills in programming, database management, analytical thinking, and experimental design. Interdisciplinary training is key, exposing students to a diverse range of topics and tools to prepare them for careers in omics. The image shows a modern computer lab setting where students are learning bioinformatics and omics data analysis, with an instructor guiding them. This slide outlines the key components of an effective omics education program to develop the next generation of omics researchers and practitioners. Public databases like GenBank, Gene Expression Omnibus, Protein Data Bank, and metabolites provide a wealth of omics data for researchers to access and analyze. Analysis platforms such as Galaxy, Bioconductor, BaseSpace, and Cyverse offer powerful tools and workflows for processing and interpreting omics data. Large-scale research networks like the Human Microbiome Project, ENCODE, GTEx, and the International Cancer Genome Consortium generate and curate extensive omics datasets that can be leveraged for a variety of research projects. Together, these public resources and analysis tools form a robust ecosystem to support omics research across disciplines. Getting started in omics requires building a strong foundation across several key areas. First, you'll need to master the basics of molecular biology, statistics, experimental design, and introductory programming. Developing hands-on technical skills is also crucial. Practice using tools like R and Python, explore bioinformatics software, and analyze public datasets. As you gain experience, you can start to specialize in a particular omics field that interests you, join research teams, and attend workshops and conferences to connect with the broader community. The omics revolution is transforming our understanding of biological systems, revealing unprecedented detail about how they function. This is enabling more precise, customized treatments and interventions, tailored to individual needs. The omics revolution is also creating exciting new fields and applications, opening up many opportunities for innovation. Looking to the future, this technology is moving us toward a more integrated, predictive model of biology that will shape the years to come.